So for this part, what we're going to get into is talking about how the ribosome has its specific role in translation. And this is going to be a continuation of the notes that we had started in class last time. So just to sort of set the scene, know where is this happening, or where is translation happening, we first have to start about where transcription occurs. So transcription, where DNA is being made into mRNA, it's going to happen inside the nucleus because that's where the DNA is located in eukaryotic cells. So once that mRNA is made, it's then processed into pre-mRNA, and then it's made into mature RNA. So there's a whole suite of things that happen, a whole list of things that happen here. If you want a little bit more detail about how that occurs, you can read about it in the book. But for today, we're just going to talk specifically sort of bird's eye view of where this is all happening. So once we have that mature mRNA sequence, it's going to be this 5' to 3' region. And because the nucleus is made up of a phospholipid bilayer. So remember in that chapter, we had learned that large molecules or charged molecules need some access out. Well, in this case, this polymer of nucleotides, which is the mRNA, is being released into the cytoplasm through this nuclear pore. So we have our nuclear pore here, and our mRNA is being released out. So then this is where translation occurs. So translation occurs inside, or actually outside of the nucleus, outside the nucleus, but inside the cell. Oh, that's pretty funny how my brain went with that. But we're just going to say that translation is going to happen in the cytoplasm, which it does because it comes through this nuclear pore. So down here at the bottom, we have our mRNA. And in this mRNA, just to get you oriented with this strand, we have our 5' to 3'. And then each one of these colored boxes is a representation of the codons. So each one of these would be a sequence of 3 nucleotides that gives off an amino acid, which is also a codon. So in this case, I just drew them as boxes just to make it a little bit more simple. But you'll see a little bit more detail in the following video to sort of wrap this all up, to bring this all together. Okay, so the ribosome, there's different parts of the ribosome kind of just floating around in the nucleus. There's a small ribosomal complex, and then there's a large ribosomal complex that are kind of floating around in the nucleus. And how it works is that these two complexes have to come together, sort of sandwiching the mRNA between them in order for it to work. Okay, so with that in mind, we have our start codon, which is this first codon here to the left side. And at this start codon, you would have that pocket of the P site that forms. But when there's a start codon here, we have that small ribosomal complex that binds to this space. So this here is going to be our small ribosome complex. So once that small ribosomal complex binds, it binds to the mRNA, it's going to recruit the large complex. So then the large complex is going to bind, and when that large complex binds, it's also going to form these other pockets in it, which then is going to become the A site to the right of the P, and then the E site to the left of the P. And this large part here, this is going to be our large ribosome complex. So now you can see here that the small complex, large complex comes together with our start codon in the P site. And now this complex, which forms the full ribosome, can start doing its job. So its main function as a ribosome is that it's going to synthesize the polypeptides, which then will form into protein. Okay, so how that happens is that it starts with the elongation. So first really quick, where the small complex and the large complex comes together, that is called the initiation site. So I'm just going to put one, and that's this initiation site here. So the next step is that this elongation step happens. And this happens where the ribosome is going to start recruiting these molecules called tRNA in order to bring those peptides to it, or those amino acids to it. So my tRNA, I'm going to kind of draw them sort of like a little hat here, and it's going to have this bead here, and this is going to be my this is going to be an amino acid. So this whole piece here, this is called a tRNA, transfer RNA, and what it does, its job is to bring this amino acid to whatever it needs next. So at this point, this one, when we have that AUG recruited here, this amino acid has already brought its complex to this point with the AUG here. Okay, so that one's already brought it there, and we have our peptide forming at this P site. So then what happens is, you can see to the right of the P site, there's this, in the A site, this is where a tRNA will bring an amino acid to the ribosome. So in this case, we have this purple one, and we're going to have a new amino acid that's going to bring the proper amino acid to it. So this one's going to be the purple. So let me go ahead and get the purple one in there. So this purple amino acid is going to bring this tRNA to it, or this tRNA is going to bring the amino acid to this space here, to this A site. Once it pops in that point, then this amino acid with the start codon is going to get shifted down. And then once it gets shifted down, it's empty. And then once it's empty, it gets released. So now we have the tRNA that doesn't have that peptide attached to it. But it's free to go grab another amino acid that's going to be given for this AUG. So it goes off, and this is called an uncharged tRNA. The tRNA that is carrying an amino acid, this is called a charged tRNA there. So as this happens, it's elongating, which means that this ribosome is just going to start shifting its way towards the stop codon. And as it's doing that, it's going to be building this peptide. I know it's a little bit hard to see because I'm drawing this out, but if you watch this video following, it will kind of piece it all together. So at this point, we have elongation where the peptides have been, or the charged tRNAs have been bringing the amino acids to each one of those sequences. And then we get to a point where now we have the ribosome that has a stop codon, the stop codon in the P site. So let me go ahead and draw in the ribosome. And at this point, because it had been building this all up, we are going to have these different colors of amino acids. So the first one's going to be black, because that was the first one that was added. The second one's going to be this purple. Next one's going to be this red/pink. And then this next one's going to be blue. And this next one's going to be green. So now we have all of our amino acids in there. We have 1, 2, 3, 4, 5. And we have 1, 1, 2, 3, 4, 5. So this is our chain, our polypeptide chain. But what happens at this point is once there is once we hit that UGA or UGA codon and this P site, there's this release factor that gets recruited here. And that release factor, what it does, this release factor gets recruited to this A site. And once that happens, then the polypeptide gets released. So this one gets released. Actually, let me draw it the other direction. So this polypeptide gets released. And then this small, not necessarily small, I will say more so the ribosome complex falls off. So what happens is once that release factor ends up in that P site, then the polypeptide is released and then that ribosome complex falls off. So then it becomes, again, the small complex. It becomes a small complex and the large complex of the ribosome. And that ends with the termination. And after that happens, then it's free to go find another mRNA and start the process all over.